I think many people agree, many scientists agree that they have hands. There's some people.
So in other words, we are going to piss off a bunch of biologists.
Some people, yes.
Well, sorry, biologists. Hello, everyone, and welcome to OpenCV Weekly Webinar, episode 61. If you can believe that, I am the co-host with the co-most, your plus one and only, Phil Nelson, filling in for Dr. Satya Malik, who is out today. In his stead, though, lucky you, we've got Anna Petrovicheva. Go ahead and say a little something, Anna.
Hi, everybody. Happy to have you here today.
And we are happy to have you here, Anna. Our guest today is Alexander Mathis, a professor at the EPFL, working on the awesome software library DeepLabCut. See, I said it right that time. First, before we get started, though, I'm going to remind you of a few things that we do every week here on OpenCV Weekly Webinar, one of which is a special giveaway to you in the Zoom audience. If you're not in the Zoom audience, you can get there by going to opencv.live. That'll be later in the episode, so you've got time to join us. I will be asking a trivia question. The first person to answer it correctly will win some DeepLabCut swag and a free OpenCV course of their choosing. So stick around for that. We'll also be doing Q&A from you in the audience. So please use the little Zoom Q&A button at the bottom of the screen there to ask your question at any time, and we'll try to flow it into the conversation. We'll also try to save time at the end to get to questions that we didn't get to during the episode. Without further ado, our guest is Alexander. It is your show.
Well, thanks so much for having me.
Welcome.
Coming from Geneva, where it's really hot these days, so I'm a bit sweating already. Maybe it's also the pressure I'm feeling from the interviewees, but let's see. Anyway, so I'm sharing my slides. Can you see them?
Looks great.
Okay, perfect. Yeah, so today I'm going to talk about Markerless articulated pose estimation with deep learning and how it's used in biology. And for this 61st webinar of OpenCV, I'm really excited I thought that I will talk about three specific kind of questions. And let's see, my slide advancer actually works. Yes, it does.
It sure does.
And so the first question is, what is articulated pose estimation and why is it important? The second question is, are there any principles that we have learned when we worked on DeepLabCut that we want to share with the OpenCV community, which is obviously very much interested in making models to do computer vision? And then I will also talk briefly about how you can use DeepLabCut. So let's dive right in. And I also want to say right away, right off the bat, that what I'm going to talk about is really not just my work. It's a work of a lot of people. So I should highlight kind of three groups. On the one hand, the experimental collaborators. You will see some exciting animals and work on those. And they're listed in the lower bottom. Most of these collaborators were actually from Harvard and MIT, where I spent a few years before I started my lab in Switzerland. And then there are a lot of DeepLabCut collaborators that are listed. And I think one that I should particularly highlight is Mathis, who started this all out with me now almost 4 years ago. And then I also want to highlight a few lab members to the right. And Jan Zuckerberg is actually funding a lot of the software development. So I should also thank these guys. Anyway, so with that, let's dive right in. What is articulated pose estimation and why it is important? So as you all know, biologists want to understand, let's say, how animals work, how the brain controls the body, how animals interact, and all these kinds of things. So animal behavior has also been studied for very long times. And I think humans were always a bit annoyed and upset by the limits of our own senses. For example, our eyes, they are not the most fast processing systems out there. We cannot hear all audio spectra, so we cannot listen to bats, for example. So people have always tried to come up with really novel sensors to better understand what is happening in the animal kingdom. And obviously one fantastic sensor is a camera. So with a camera, you can really nicely capture animal behavior. And I think to people, I mean, the OpenCV community is not the right audience to say this to, but for other people, maybe it's not, it sounds easy to analyze data from videos a priori. But as a matter of fact, machine vision is a very difficult topic. And one of the reasons why OpenCV exists is actually to do machine vision and to do it openly and together. And so
yeah, I think we definitely would not be nearly as popular if it were easy.
Exactly. So another issue with videos is, of course, that this video, no, it has a lot of pixels and it has a lot of bits of information. So depending on what you want to study, it might not have, like, it doesn't have a condensed version of whatever you want to study. So how should we basically compress this kind of information to understand behavior? And historically, people have kind of realized that let me just show you a different kind of representation, and then you will see maybe a good motivation for pose estimation. So unlike this previous video, what you see here is just a few dots that coherently move over the screen. And this is actually from a very old and very classical video by Gunnar Johansson. You can also find the YouTube link. It's really something I recommend because there's much more in this video. But what is, I think, remarkable is when you look at these few dots moving coherently, you look at them and you cannot help but see humans walking, humans shaking their hands, humans hugging. And that is to say that this kind of very low dimensional representation of what these two individuals are doing is a great way to summarize behavior and to capture behavior. And for that reason, one of the big goals in computer vision, essentially, actually, for many downstream applications is to go from high-level pixel representations, so think of this video of zebras, to low-dimensional key point representations of the key points of interests that you want to, let's say, track. And in an abstract mathematical way, this going from the pixel representation to the key point representation, that's what is called a pose estimation algorithm. A pose estimation algorithm typically will not just find in X and Y coordinates on the target image the key points of interest of all the individuals, but it even will group it by individual. So you will have subject 2, let's say in red, and subject 1 in blue here. And let's say, like, before I talk about how this is done in practice and a little bit of the history of how kind of pose estimation was done, I also want to highlight what can be done with this once you have the data. So as you can imagine, once you have kind of key point estimation data, you get these like low dimensional time series of movement of animals, let's say, or of humans. And what you can do with this is you can then cluster them into actions, what behaviors are happening. As we saw earlier, it's easy for us to actually classify that they are shaking their hands or walking or whatnot. So, or you can estimate kinematics. That's important, for example, in biomechanics. You can then also correlate it to brain activity. So what you see up here is actually recordings from a mouse brain, and down here is pose estimation data of how the mouse is moving. And people are really interested in, okay, how do these internal units of the brain create this behavior, or how do they encode it, how do they decode it, and so on. And then people also do a lot of biomechanics. So these are just a few applications of pose estimation. But but what I now wanted to talk about, I hope I motivated pose estimation sufficiently. Otherwise, Phil, ask me.
Yeah, one of the things I would like to mention here is I love how that first representation on the right side is very evocative of like ancient cave drawings. It looks very much like things you find in, you know, prehistorical times. Mm-hmm. Of the representations that people carved into walls, and we're just kind of doing the same thing here, but with sand that we taught to think. So yeah. It's kind of
and totally. I think kind of the skeleton is like an abstraction, like the essence perhaps of movement of an individual. And so I guess, yeah, people saw this a long time ago and drew it in caves. Anyway, people also were like, historically, people also wanted to go to this key point representation, but it is really, really a hard computer vision problem until this day. And the easiest way to kind of deal with this problem is to make it easier by hardware tricks. And so what I think the earliest contribution to Markerless, marker-based pose estimation is actually by Bernstein. So what you see here to the left is a Russian worker with a hammer, hammering. And this is a picture taken by Bernstein, where essentially what he did is he attached light bulbs to 3 points on the body of this, on the body and on the tool of this worker. And then he took one picture. And because the PIC camera was slow and it integrated the photons, you basically see and can analyze the trajectory of movement. So this is obviously one way where, well, through photography and hardware, you can analyze the data without any fancy algorithms. And this technique of doing marker-based pose estimation is really has started there and is very common. And I want to highlight one study here to the right, which is a neuroscience study. So you see this monkey actually has an implant in motor cortex, and there's this block in front of it. And it is a beautiful task where they actually study how the monkey controls its arm. And in order to get the monkey to move to lots of different locations where the monkey can actually reach to, what they do is they swing this block at the monkey, and the monkey is just to catch it out of the air, which they can obviously do very, very well. But even though this was in 2010, in order to understand how motor cortex controls all these muscles, there's more than 30 muscles in the arm of a monkey, what they wanted to do is they wanted to very finely measure all the degrees of freedom of the arm. So what they had to do then was essentially they shaved the monkey. So you see that this monkey shaved, and they put these markers on the monkey, which the monkeys were not very excited about. And when you talk to the authors, and I know these guys, they told me that it took them actually half a year to train the monkey to accept the markers, because, well, it's really not like they weren't into this. And the study actually had 3 monkeys, and there was one monkey from which they could never record data, because always when they had rewarded him for putting all the markers on, the monkey would just move them off and then say, okay, I'm done with this task.
I'm very, very proud of that monkey.
Yeah, I think he's a real winner. He knew really how to do the task properly to get all the reward for putting on the markers, but then just stop working. Yeah, so for that reason, people, especially in the animal field, were always interested in marker-free variants. And people have also worked excessively on marker-free pose estimation methods. But I think essentially the situation changed a lot when deep learning came around. And so here I put down a bunch of algorithms. I think the first algorithm for using deep learning for marker-based pose estimation is actually from 2014. And then in 2017, there were 2 papers, one of them that inspired the name of DeepLabCut called DeeperCut, and another one, OpenPose. These were actually the first papers, I would say, that were really fast and could do multi-human pose estimation. And so to the left, you actually see a video of OpenPose. So what you see is a bunch of humans dancing on a pier in Sydney, I believe. And this algorithm predicts accurately, very nicely, the key point locations of all these individuals. And importantly, it hasn't been trained on the scene. Oh, like it just works basically out of the box really, really well. And so what's the principle of these algorithms? In a nutshell, like in prior algorithms, the idea is you have an image, a frame from your video, you feed it through a predictor, predicts a pose. And the major difference here now is that the predictor is actually a deep neural network with lots of, in this case, it's actually a convolutional neural network, lots of layers of convolutions. And all these convolutions are end-to-end trained with a lot of annotated data. So this is actually trained on COCO, which is a large-scale human benchmarking data set. And with that, they trained the models, and then these models generalize actually pretty well to such situations.
Was recently talking with a frequent guest on the show, Joseph Nelson, no relation, of RoboFlow. And one of the episodes he's been teasing for a while is called Death to Coco. So we'll, you know, keep on the lookout for that, everybody. I wanted to interject here a little bit. Yeah. I know, Anna, you have a lot of experience with pose estimation and people tracking and stuff. Is there anything you'd like to kind of add to this historical look at human pose estimation?
Yeah, I mean, this is probably like super close to my timeline of work, because I also was deeply following these berry papers. This very video actually surprised me a lot when I first saw that. I wanted to like 100% second Alexander saying that like this really changed the industry, right? No kidding. It's not only a fun video to look at, it actually unlocked so many different businesses that are thriving right now or only starting to kind of unfold. This is a really, really big thing, even if it like, seems fun.
Yeah. And I think it's, I mean, if someone wants to appreciate that more, then just try to do it without deep learning, even nowadays. I think it's really hard to get to work.
I mean, I tried.
Yeah.
So my computer vision career, my computer vision career started before deep learning. So I first was struggling with this, like to make these, very simple filters work, and then ta-da, it works.
Yeah, exactly. So, and I will actually show some data later that I have worked on, and where I tried to actually use OpenCV and like classical, like, thresholding and whatnot, and ellipse fitting, and it actually just didn't work. And it was quite a nightmare. That was roughly in 2017. And then I saw this video and I'm like, okay, someone can do this, and I cannot track the snout of a mouse in a black and white box. What's happening?
I think we've all asked ourselves that question at some point in our lives. So someone is doing this and I can't track the snout of a mouse. What am I doing? Just, that common thought.
Yeah, at least for neuroscientists, let's say. So yeah, with this, so for humans, this works really remarkably well. I mean, there are still some problems, obviously, that we can discuss maybe later, but it was really a game changer, as Anna is saying, and it works spectacularly well for many, many settings. And so for us, what we wanted to bring this to animal pose estimation, essentially. And there were a number of kind of challenges that we identified for bringing these things to animals. So the first challenge is, okay, it works really well based on COCO, because you have a lot of annotated data and you can create a model that works well for humans. But do we have to create tens of thousands of frames that we annotate for each animal in order to make it work? Which maybe is a bit of a problem, because you probably know how diverse animals look. They're fruit flies, they're monkeys, they're cheetahs, they're fish. So given the diversity of animals, it would really be tricky, and the diversity of body plans, to maybe create these large-scale data sets. And then people also wanted to have, of course, sufficient accuracy for doing biomechanics studies or neuroscience studies. And another thing that was super important is that we want these algorithms to be extremely fast, because one thing that people want to do is they, for example, want to give closed loop feedback to an animal based on what they're doing, in order to see how animals learn, or how their brains control what they're doing. And I also put multi-animal pose estimation and tracking as a challenge. And the reason here is really, I mean, it is extremely impressive what you see in the previous video, but keep also in mind that all these people here, they look quite different because of their different clothes. But imagine there would be 20 mice dancing. They all look highly similar. And so these are a little bit of, I think these can be some challenges. And, or in particular fish. I will show a fish video later where it's becomes very apparent, this problem. Anyway, so now I'll dive into DeepLabCut now a bit. And I will do this with kind of this goal of actually sharing, I think, some principles that we have found and that I think are useful for people to train neural nets on doing various tasks. So kind of on a personal note, it was indeed the case. So I was working on this olfactory behavior, and I wanted to track the snout of a mouse, and it just actually didn't work for the longest of time. And Mathis, now my wife, she was working on mice. So she had trained mice to pull a joystick and play computer games, essentially. And she wanted to understand the articulations of their hands when they pull a joystick. And also that was a very hard problem. You can imagine the hands of mice are really small. And by the way, I say hands because they don't have paws, they really have hands, okay? As you can also see in these videos, they really have nice 4 digits. They don't have a thumb, they have a claw. but they do have hands. And so what for us, essentially, the surprising result was that if you happen to, let's say, have a bunch of frames that are very different, where the mouse hand is in different postures, and you literally click on these different frames and you annotate them with the data that you are interested in, then if you enter and train a relatively simple architecture, which is actually was a subset, or is at that time is a subset of this algorithm called DeepLabCut, which is simple in the sense that it's really a ResNet backbone. So this is a very, very powerful deep neural network that is widely used. I think the paper has more than 100,000 citations now. So it's a 50-layer CNN that has these skip connections, which are really useful because with these skip connections, you can train it out very deep. And at the end, it has these deconvolutional layers. And what the algorithm actually does not do is it takes basically this image and predicts the X and Y coordinates, as in regression, of where the key points are, but it actually does it like an image-to-image translation problem. So it takes in this image, and what it seeks to predict is for each body part, a probability density, like a little density, where it says high probability at the location where the human annotated and low probability where it didn't. And the exciting thing for us was that if you end-to-end train this architecture, when it's pre-trained on ImageNet, I will talk about this in a second, then even if you label 141 frames here for this specific case, and you run this on a new mouse, a different mouse that runs is part of your study here, you see the joystick, by the way, and the mouse pulling the joystick, it works really well. Yeah. And I think for people that have only seen deep learning, that's maybe not really impressive. But I think for people that have tried to make such trackers before deep learning, this was very, very surprising. And especially that it's so little data that you can actually use now. And
so I've got a question here, which is exactly what video games was Mackenzie teaching them to play?
Okay, that's a good question, but that's a whole other talk, of course. But essentially, she was teaching them video games where she would, so they have to learn to move the joysticks. And once they're experts at moving the joysticks, she would introduce tiny forces and displace the joysticks unknown to the mouse in order to see if they can learn an internal model of these external forces that apply to the joystick, which they can actually do, and how the brain essentially does this. That's kind of the short answer.
Wow. That's very interesting.
You know,
again, this may have to be another episode of the webinar. Go ahead, Anna.
Yeah, definitely. We need your wife on our show. That's 100%. Like I saw deep learning, but I'm super surprised looking at that because they really have hands. These are hands. This looks a bit scary to me.
Are we going to piss off a bunch of biologists, or is it a solved argument that rats have and mice have hands, or just rats, or just mice?
I think many people agree. Many scientists agree that they have hands. There's some people.
So in other words, we are going to piss off a bunch of biologists.
Some people, yes.
Well, sorry, biologists.
But many neuroscientists and biologists totally agree that this is not a paw, as people would naively think, let's say.
That's really interesting.
Yeah. Great. So yeah, bottom line there was, with relatively little data, you can make this work. And that was really surprising to us. And the second thing then we did is, okay, so now we wanted to understand, okay, how well does this really work, and how well does it maybe work in comparison to ground truth? So it's of course hard to get ground truth annotation data unless you do X-ray, let's say, which is one way. But so what we did is, and this is, by the way, this setup that I was working on. So here you see a mouse from above. It's actually running on a treadmill. And I printed, so in this setup, we print odor trails on this ground, on this paper. And the mouse is following these odor trails because obviously that's just very exciting to follow like smelly trails on the ground. And what we also do is actually we printed chocolate milk. And because as soon as they realize that there's chocolate milk that they can drink, they wanted to obviously follow these auto trails. And so what we needed to understand is how well can a human actually do this task? And so what we did is, so we had one guy on the paper, Pranav. Actually, everyone did it, but Pranav was spectacularly good. So he annotated 1,080 images once, and then more than a month later, because we wanted to discard memory effects, he annotated the same 1,080 images again. And what you see here is exactly that. So you see in blue, the first annotation, and in yellow, the second annotation. And often it just looks green, which means blue and yellow overlapping looks green. Sometimes there's some displacement, and then here are these numbers. So here, for example, he was 1.55 pixels accurate more than a month later. And here's his whole distribution of errors. You see that very rarely he has big deviations. And on average, it was 2.7.
Get this guy to Las Vegas and make some money.
This guy is he's probably one of the best laborers. He's also really great neuroscientist, to be very clear. I should say that. Okay, so now we knew how accurately a human can do this. So now we were wondering, okay, and by the way, to give you an idea what 2.7 pixels is, here is 10 pixels. This is the scale above 10 pixels. Oh, sorry. So this is really not much. So now what we wanted to know is, okay, can you actually reach human level accuracy first? So for that experiment, what we did is we took 80% of our data and we trained a model, actually several splits with 80% of the data. Then obviously your model will overfit and actually will be even lower than human variability. So here where you see the mean square error difference between the prediction and the ground truth. And then in gray, that's the human variability. And you see that actually on the rest of the data, these 20%, the model actually reaches human level accuracy, which is exciting. And just based on, let's say, 800 images, which is not that much work either. And on top of that,
this is a really good graph, but I think some of the audience may not be familiar with RMSE as a term. You want to maybe talk a little bit about that? So you're kind of describing it here, but
yeah. So it's just root mean square error. So think of it like the Euclidean distance. So for a given body part, you would just take the Euclidean distance, which is like the shortest distance between the two pluses in 2D space. And then you averaged it over body parts and over only body parts. And then that's root mean square.
Right on.
That makes
Thanks.
sense. And so then when we varied the amount of training data, and even if we went to relatively little, like just 50 training images, then the test error of the model on average was still below 10 pixels. And remember, this is 10 pixels. So this is really a usable model to kind of predict key points. And so so what is also important here is this is really useful for biologists, depending on, let's say, Mackenzie, she was interested in reaching. So she could annotate the key points on the hands and track that. I was interested in tracking the snout. I actually got a model that could track even more body parts than just the snout. And it could do the snout really, really accurately, which was super useful for me. But the kind of higher level computer vision principle from here is that if you start with a ConvNet, such as ResNet, that is pre-trained on this large scale data set called ImageNet that you probably all have heard about, or several of you have heard about it. So it's an image recognition task that has millions of images and 1,000 different classes. Then you can take this backbone, so this kind of neural network, which at the end has some decoders to predict the classes, and you can transfer this main part of the net and you put a small decoder and then train this model to do essentially pose estimation. Yeah. And so this is what is called transfer learning. The name actually comes from biology because it's based on this idea, okay, if you know some tasks and you want to do a different task, then you don't start from scratch in order to do these tasks, but actually you transfer some of your knowledge over to this. And this is really something that is happening in what people do in computer vision, and it's extremely useful because you need much less data and so on. So the kind of, I think one really nice takeaway here is, so this, I took this picture from Andrew, and you see that people always had learning algorithms and were very interested in learning algorithms. But in this caricature, what essentially the problem of all the learning algorithms is they just hit the wall at some point. And even if you throw more data at them, they maxed out. And so what Andrew was kind of making a point is that with deep learning, it's amazing. You can just build a big architecture and so on, and you can always get better and better. But the problem is that, like, if you look at this area here, then maybe all the learning algorithms would actually generalize better, perhaps, if you have small data. And for most of us, because we're not Google and Facebook and so on, we don't have large scale data available. We are maybe more in this regime. And so I think in that regime, transfer learning is kind of one of the key tricks, because you can essentially get a really good performance with relatively little task related data, because you basically exploit other data that you haven't annotated specifically for this problem. Okay, so that was one of the big take homes that I wanted to share.
Yeah, Andrew's been talking a lot about data as it relates to AI and deep learning and transfer learning and all this stuff a lot over the last couple of years. And it's definitely, a lot of the folks that have been on the webinar have talked much about how there's not a one size fits all solution for data, even in terms of how you capture the data and how much you use. Task to task, it can be completely different, and that's good. It's good to curate and specify data for specific tasks. I think that's a really interesting and important point, because people just think, oh, we've got ImageNet. There's millions of images. We don't need anything else, and that's not really how it works. And
no, ImageNet will generalize poorly to other object recognition tasks that are very different in distribution of data in classes and so on. So in order to actually address specifically this question, so far the data I've shown is really kind of relatively specific lab setups where the variability is relatively small now. It mostly comes from the posture of the animals. There's a little bit of luminance changes from one day to the next. But one thing to kind of assess how well these algorithms can generalize to very differently looking scenarios is actually looking at horses. And that's one data set that we have looked at. So the data set that we've created, it's called Horse 10, I believe. It looks like this. We always have videos of horses running from left to right. The key points that we annotated are overlaid. There are more than 25 or something. And we have 30 different horses. So now these look really quite different though. I mean, obviously horses, that's clear to every one of you, but the backgrounds are different. Some of the videos are taken across the sun, against the sun and whatnot. So what you can now ask yourself is,
but do they have hands or do they have hooves?
They have hooves.
Good, yeah, good. Just making sure, just making sure.
I think here all the biologists agree.
See, there's going to be some fringe biologist out there that's going to be like, Sorry again, biologists.
Anyway, so with this data set, what we now wanted to ask is the following question. We wanted to ask, okay, how well, so we wanted to test this generalization question. Namely, if you happen to have only the data of those 10 animals here to the left, that's why it's horse 10, although there are 30, then you can ask yourself, okay, now if I train a model on some of the data from these 10 animals, then presumably it will work on these 10 animals. And the answer to this is yes. But you can ask yourself, okay, how well does it generalize to these other 20 animals, which some of them are similar, some of them are not. And by the way, this is a benchmark that you can compete on if you want to. And we found something quite interesting, and I want to walk you through this because it's also maybe a complicated plot. Namely, on the y-axis here, that's kind of normalized error. So that's, again, it's kind of the same like RMSE. How well can you predict the body parts? It's normalized because, let's say, you can see to the right here, some horses are big and some are small. So 10 pixel difference might mean very different things for a horse that is as far in the background as this one, or that is closer in the foreground. Yeah. So now that's why
scale is such an interesting problem, no matter where it is. 2D, 3D, pictures of animals, pictures of buildings, scale, always a problem.
Exactly. So that's why it's normalized. And that is actually for neural nets, that's easy to deal with, no? Because when you train them, you just augment them with scale augmentation. Like you kind of scale the image plus or minus whatever the range it will encounter in its deployment. And then you train the model that way. But now what you see here is firstly the statement that I had that if you look at the within domain, so for the 10 same horses, then the test generalization is really good. That's the first thing you see here. And this is, by the way, for ResNet 50 that I've talked about most. And what you will also see here is that actually this model will not generalize so well, but here's how well it generalizes to these 20 other horses, okay? So let's keep that in mind. And then one could do something different because, I mean, strictly speaking, I've told you, oh, transfer learning is so amazing. We can get away with very little data, but I haven't shown you that. So now what could we do instead of transfer learning? We could say, well, we have this CNN with some decoder. Can we just train it end to end on our data? And as a matter of fact, that works if your data is big enough. And in fact, if your data is big enough, then you will most likely match or slightly surpass. There's a really exciting paper from Kai-Min He, the performance based on transfer learning. But there's one thing that is interesting here in this plot. So we still stick with this ResNet point. Namely, if you train it from scratch, so you randomly initialize your whole big model and then you train it on this task, it actually generalizes much worse to these 20 novel horses than if you start with transfer learning. So transfer learning is not only good for you requiring less data, training faster, but it also really helps you to have a model that is much more robust at generalizing to other data that is somewhat similar, but not too similar, perhaps. So I think in practice, that's also super important. And so far, I've only talked about the ResNet part. So this is just one model. But there's one other interesting question that we were after and that we thought is quite interesting. And it's this question, if you have a model that is better on ImageNet, so it's better at doing object recognition, will it also be better at doing poses estimation? And so what you see here is different backbones. So for example, mobile nets, ResNets, efficient nets, and they actually have different performance on ImageNet. And what you see in this plot is that they are somewhat correlated on the train and test set, no? Like the normalized error really goes down with a better backbone. And so what is interesting here, there's nothing different otherwise. Like the algorithm, the data is exactly the same. So the only thing that is different is this big deep neural net in between. But if you start with one that is basically an efficient net, which is better at the image recognition, then we'll generalize actually better also to pose estimation. So I think one take home here is that maybe if you have an application where you want a model that is very robust to a lot of diverse data, then it is better to take a backbone that is really big, which of course will come at the cost of being slow in comparison, because mobile nets you could run on your cell phone, let's say, easily, but an efficient net, maybe not. So that I think is another highlight that I wanted to make. And the same is, by the way, true really also for the out of domain data. So on the out of domain data, there's also this, so really better models on ImageNet really generalize also better to novel data. And so I think that's quite useful. And by the way, you might wonder, okay, is this only true for generalizing from these 10 horses to these 10 horses, or is this a general principle? So we have done a really fun experiment in this paper. You can read more details in this back.
Those are some odd looking horses.
Yeah, this one, it is an odd looking horse, yes. So here, what we did is we asked, what about generalization from one quadruped or from some quadrupeds to others? So we hold out, for example, the sheep at the bottom here or the cows, and the same is true. So kind of this generalization principle is not just specific to a bunch of different horses, but it's really the deep neural net probably better understands where the key points are on a quadruped in a case when it is better on ImageNet than if it's worse. So that's what you see here to the right. Yeah, so are there any questions about all of that so far?
Yeah, I have. So one of my questions for what we just covered here specifically was, what You didn't really talk much about your preconceptions before you started work here, and so I'm interested in how these results have surprised you specifically.
Yeah, I mean, I did talk a bit about that. I tried to analyze my mice in a box, and that was a much more difficult process than I thought it would be. And what was really amazing is once we tried deep learning methods, they just worked so well, and like shockingly well, actually. To like, certainly I was deeply surprised by how well they worked, actually. And yeah, and then we wrote up the paper and we open sourced our code, and our first tweet actually blew up completely. The next day there were GIFs from other people analyzing their data, and we were like, oh wow, okay, this really does work. Not
Yeah,
just
that's in fact how I saw it. It was I believe it was retweeted onto my timeline by Kat Scott from Ross. So thanks for that, Kat.
Yeah.
Here's a question.
Mm-hmm.
Here's a question from me. So you mentioned that like we as a people, we look completely differently, but mice, they look more or less the same. And I read that, you know, like in some cases, a person cannot kind of distinguish mice, but the mice themselves, they distinguish each other pretty clearly. Do you see effects like that when training a neural network to distinguish mice? Because my suspicion is that we have some hardwired mechanism to, you know, distinguish people because we are people, and they should have some hardwired mechanism to distinguish mice, but maybe a neural network is able to learn that too.
Yeah, no, it's a fantastic question. So I want to, so there are a lot of things in your question. One thing that I want to emphasize is, so humans do look very different, no? But I think, but it's also clothes that make us often look even more different. Like they add a complete other layer of diversity that, let's say, you don't have in mice. And there are different mice, no, some that are white, black, and whatnot. And that said, so there is really no evidence that mice can visually identify the identity of a mouse, I think. I have tried to find that paper, but I don't think there's really evidence for this. I don't even think that there's very strong evidence that mice can discriminate in the, like, individuals a lot, like very well, apart from, let's say, their parents or something. In fact, only their mother, I believe, but don't quote me on this. I'm not an expert on that. But I do think that mice maybe can do this. Like if they can do it well, they most likely do not rely on vision, but they probably do it based on sound and based on the sense of smell, which yeah, and that is similar for many other species, you know, like dogs and so on. They can tell each other apart based on smell, something that I think humans are not so good. Yeah, and one fun anecdote, by the way, about, so I think that one of the species where it's best understood how well they can identify each other is penguins. And I think there's even the mechanism is understood. And I think, so these king penguins, they can find their chick in a group of more than 100,000 individuals based on their calls, which is, I mean, that's remarkable.
Oh, wow.
Yeah, that's such a huge number.
It's such a complicated, like, decision-making task, no? Like, yeah.
Wow. Alexander, are there more slides, or are we just in?
There are some more slides. Yeah.
Okay, yeah, let's continue with the slides here then.
Yeah. So yeah, so one thing that happened then, actually, like sticking a bit with the personal story, I think a few weeks after we had our paper on arXiv, I got this email from a person from Amir Patel in South Africa, who was working with cheetahs. And he said, oh, do you think this algorithm will work on cheetahs? And I had actually absolutely no clue. But of course, I think like everyone, I always wanted to work on cheetahs, because I mean, who doesn't? So I said, yeah, let's collaborate. Wonderful. So he sent, and he said yes, and we did. And this is actually his data. So here's a video at their range in South Africa. You see a cheetah running.
Blink and you'll miss it.
You don't see it.
Blink and you'll miss it. It's just so fast.
It's quite a fast animal. And what they have is they have 6 cameras here. But the first thing, so you see actually 3 on the other side, and then there's 2 more to my left, I guess, somehow. So indeed it works, no? The same algorithm works. Like if you annotate enough data, it can do a really good job on these highly agile animals. And now, and in fact here, I should say we train actually one model for all the cameras, because I mean, from the different perspectives, it looks pretty similar. And you can, it's just much easier to keep track of that. And then once you have all the 2D data and you calibrate your cameras, what you can of course do, and by the way, this is also partially based on, of course, on OpenCV, you can do 3D pose estimation. No? Like you take in your 2D data and you create 3D estimates. And here there are 3 different methods shown. One is triangulation, which is, I mean, it's well known that it's not very robust if you have outliers in 2D. And then there are 2 other methods. And then this is the 3D reconstruction. Yeah. So I think that's great fun.
So cool.
And yes.
Extremely cool viz.
Yeah.
We need more episodes of the show with giant cats, by the way. I need to work on that. And
I'm realizing there's no, here. If you want to read more about this, there's the reference down here, so you can check out that work. And there's a large open source data set available if you want to train models on cheetahs yourself. It's called AsinoSet, and it was actually published at this robotics conference, ICRA, last year. And so with that, I want to kind of get to kind of a little bit summarize here as an intermediate conclusion, essentially. So yeah, it was really quite an exciting journey. We have been published quite a number of papers on various extensions since 2018. And because now it's based on general deep learning principles, it can be applied to really a lot of different things. Like it should no longer surprise you that you can apply it to mice, parenting, or 3 mice in this box, or this mouse actually running on a treadmill. But maybe it's a little bit surprising that you can apply the very same algorithm to actually analyze T cells. So T cells in the immune system killing cancer cells in these wells. And what the algorithm here actually does is it just detects the corners of each well automatically, left lower corner, right upper corner. It automatically detects the different T cells, the different cancer cells. And because when the cancer cell is dying because of the T cell, it changes its color. We can also detect that it's dying, that it's a lysed cell. And so you can read about this in this paper. So yeah, the code is all open source available. It has been now downloaded more than 350,000 times, which is nothing against OpenCV. I know that's 1 million per week or something.
For us, that's like a Thursday, yeah.
That's still, I mean.
Yeah, we were super excited about it, and it has, I think, quite a nice and lovely active user community. We have more than 80 contributions on GitHub, and I think it's really used in lots of labs around the world. And
that's so great. And again, this is all sprung up in such a short amount of time. To see that sort of adoption and such a broad community so quickly it has to be really gratifying to see this work that you spent so much time on explode like that.
No, absolutely. Yeah, it has been really gratifying. And so then now for the end, I just have a few slides on kind of to give you maybe a better idea of how you could use DeepLabCut and how it looks maybe as code. And for doing that, I really kind of want to highlight what it is based on also very much. So firstly, it's really built on a lot of open source Python stack, like TensorFlow, OpenCV, of course, NumPy, SciPy, Pandas, whatnot. And that's really, really important though. Like without this, there's no way we could have ever written that software. And it's of course also built on lots of work in computer vision and insights in computer vision. It can also, because of a lot of work of a lot of other people, you can kind of use the models and deploy them on various different platforms. We have actually Docker containers, so you can easily reproduce them in different environments and run them. You can run it on Amazon Web Services. There's DataJoint, which is like a platform to do keep track of your experiments in science, which is very important. And then it also interfaces with a number of neuroscience specific tools, which I don't know how many know those, but for example, there's Bonsai, which is a really cool reactive programming language for also making like tasks for experimental work, and many other kind of post-processing, as I would call it, where people like made methods to create ethograms or cluster the data or do analysis of motor behavior and things like that. And so that's kind of the huge ecosystem that DeepLabCut is built on and really rests on, and also contributes to. And then the kind of workflow of DeepLabCut itself is, maybe as you can imagine, it's kind of a software 2.0 situation here. Namely, it integrates, like for many other deep learning aspects, and I mean now in OpenCV you can also make deep learning models. It works in a similar fashion. For those kind of programs, what you need to integrate is essentially the annotation data that you have, and maybe is task specific for yourself, and the training and the inference. And so what DeepLabCut does is it essentially, let's say in a nutshell, has 4 steps that you need to do in order to do this on your own data. So first, we call it, you create a project, which is basically a folder on your computer where you will put all the hyperparameters for your model and whatnot. You will also name the body parts that you want to annotate. And this folder will also be used for extracting differently looking frames into. Because I mean, one thing that is of course important, imagine you want to analyze mice reaching. You don't have to annotate, I should say annotate. You don't have to annotate frames where the mouse is basically doing the same thing 100,000 times, because there's no diversity in this. So you really want to make sure that you have frames that look as different as possible. And so for that, there are actually methods in DeepLabCut to extract frames that are differently looking based on like clustering techniques and so on. Then there are also graphical user interfaces for annotating the key points that you want to annotate. Then once you're done with this first part, you would select your deep neural network. Let's say you want to have a very robust one, and you take a big backbone. Let's say you want to try and run it on a cell phone, then you would take a small one. Then you train your deep neural network. By the way, you can also share these projects because they're just folders, and you can deploy them on a server, or you can train them on your laptop or whatever you want. Then once you have a trained model, you evaluate it. Of course, you hold up some data, you check if it generalizes well to that data. Let's say it doesn't, then you can refine the data. For this, there's active learning methods in DeepLabCut, and there are also graphical user interfaces that are there to kind of refine it. And then once you're happy and the model works reasonably well, you can just run inference and plot your results and so on, get your results. So all these plots, for example, are also created with DeepLabCut. So this is, yeah. And then if you now maybe want to learn more about it, there's a Nature Protocols paper where it describes the whole workflow. I mean, this is maybe more geared at scientists, but you should really think of it as like a nice cooking recipe. Here are 10 steps to get your neural network to analyze whatever data you want to have analyzed. And just to give you an idea of, so one thing that we were very interested in, I think it's also a principle of OpenCV, to have kind of function names that are really intuitive, that you don't spend your day reading kind of the documentation. But
definitely,
let's say that if you,
as a Python guy, I definitely feel a burning in my heart when I see some badly named functions that I'm like, come on. Every time I get this wrong, or like I do it backwards, so yes, I feel it.
Yeah, so exactly. So for these 16 steps now that you maybe need for doing a DLC model, they're like reasonable English names, like train the network. And guess what? If you do DeepLabCut.train network and the config path pointing to this folder for this project, it will train that model. So I think that's something that is nice. But then let's say you're not into Python so much, then there's also a graphical user interface that you can use and that allows you to do all these steps. And
is this with tkinter, or what GUI library are you using here?
it's WX Python,
Gotcha.
but we are switching.
Fair enough.
After 4 years, it's about time. Yeah. I mean, it is a nice, it does work quite nicely. There's some issues sometimes with installation, I would say. So with that, I wanted to, so I'm almost done. Time-wise, we're also almost done, so I guess that's good. But I wanted to highlight one more thing, and that's kind of a citizen science aspect. So we also, so we work with a lot of different groups doing various projects in biology, and there's a website called DeepLabCut which is basically a web-based GUI. So everything I described so far now is a Python package with graphical user interfaces you would install on your computer. But we're also integrating this now with a web-based platform. And already now there is this kind of labeling graphical user interface that you can have in the browser. And you can go there and you can annotate data for various science projects and contribute to these science projects. And you might wonder, awesome. What is that useful for? So this is being used to create models that broadly generalize across different species so that other scientists can just download these models and analyze the data without even clicking on 141 images at all. And to give you an idea what, I mean, these animals are actually there. By the way, they are cat and dog models. So if you want to analyze your cats and dogs at home and what they do when you're not there.
Or your pet giant squid or your cheetah.
Yeah, who knows what animals people have at home. Then you can do that. And I would just want to highlight one specific model that is in the model zoo. And this is from a team from Japan, Labratory et al. And so they have contributed a model that works very well actually for rhesus macaques, which some people.
Wow, that's really impressive.
Yeah. Now the term model zoo really means something, right?
Yeah, so that is true. Here, model zoo is kind of double meaning.
Yeah.
Wow. Yeah. Okay, so with that, I come to my summary. I think I was mostly kind of talking about the bit of the story of how DeepLabCut was developed and what it can do, that it's based on transfer learning, and that's really a lovely principle that helps you to make highly robust and generalizable models. Yeah, because of the power of deep learning, you can use these things in complicated lab scenes in the wild, also in clinical settings, which a lot of people use DeepLabCut in clinical settings. And you can also just use it for fun at home, of course. And then there's the DeepLabCut Model Zoo. And there are two links here, one to the kind of GitHub repo, where you can find a lot of materials, so check them out. And then there's also the DeepLabCut website where you can find tutorials, data, papers, and whatnot. And then I come to my most important slide again, the acknowledgements. And I had started with that, but yeah, thanks to all the collaborators, it has been really a fun journey.
Mind-blowing.
Yeah,
really.
Thanks so much to everybody that worked on this. And yeah, thanks so much, Alexander, for the presentation here. Awesome presentation full of kick-ass hard science. I mean, what else can you ask for from OpenCV Weekly Webinar? There is one more thing that people can ask from us, which if you'll please unshare your screen, Alexander, we will do our trivia question and giveaway. So yeah, just go ahead and hit stop sharing screen wherever that is.
I found it and I managed to hit it.
All good. So so one of the things we do every week here is our trivia giveaway. This week we will be giving away some DeepLabCut swag as well as an OpenCV course of your choosing. I am going to ask a trivia question based on the slides we just saw. Please answer in the Zoom chat window. If you have won recently, as in the last 2 months, please do not answer and give someone else a chance to win, okay? Get ready to answer. Earlier in the presentation, they talked about putting trails for the mice to follow. What substance did they use for those trails? Be more specific. Oh, I think I got one. Looks like Francis was the first one. Some of you said chocolate, some of you said milk chocolate. The answer was chocolate milk. Francis C. got it correctly, though you misspelled chocolate in your haste. I'm going to give it to you anyway. Please send one email to phil@opencv.org with your name and that you won today, and I'll make sure that you get your prize. Congratulations. I think we should at least do it. You got time for a couple of questions, Alexander? I don't know, we're a little bit over on time. Okay, cool. Lots of questions here at the coming up at the end of the presentation. Girish would like to know, what's the licensing for DeepLabCut?
Yeah, I mean, it's GPT-3. Well, no, GPT-3, that's something else. What am I trying to say? It's LGPL-v3, and it can also be used for commercial, and actually a bunch of companies that use it too, but mostly academics. But yeah,
excellent. Great. So you mentioned flies earlier. Have you tried this method with bugs, with insects, like centipedes, for example?
Yeah. No, I saw there's one question from who's Justin. Yeah, no. So even in the first paper, we also showed it for flies, and you can. I mean, it's general. No, as long as you click on the key points that you want to track, you can do it. So it has been used in birds, in fish, and even plants. It has been used in plants. It has been used to analyze physics experiments where people want to track, like, let's say, some object, because it just works quite nicely. It has been used for magic tricks. That's maybe one of my favorite papers, where people track the hands and the keys and the coins that magicians were using.
So it can actually detect when they're palming stuff, like even a really good magician,
it's a really fun paper.
A teller level magician.
So it's a really fun paper. Let me drop it in the chat.
Yes, please do. I got to contact these folks so I can have them on the show.
Yeah, that would be wonderful. Absolutely. Because so what they find in the paper is that there's some magic tricks that, let's say, humans are fooled by, but DeepLabCut is not, and vice versa. So it's really
that's really interesting. I've always been fascinated by up close magic, by sleight of hand magic. I talk about pretty often on the show. I think we talk about like 3-card Monte, or like the shell game, that sort of stuff. And so, thanks. I'll make sure this link goes in the show notes as well. Thanks, Alexander. Yes, Anna, do you have any questions here at the end of the show?
I mean, like, my question is more kind of philosophical level. So again, I'm really it's mind blowing what you do, and the impact, both in terms of the kind of hard science and the impact and stuff. I'm wondering what drove you and maybe your research group to kind of start this, right? Because I totally understand what drives you now, right? But what was the initial driver?
Yeah, I mean, the initial driver was really like, I wanted to analyze these olfaction experiments, and understand how mice were doing this task. I just had a hard time analyzing the data. That really was it that drove us to this. And Mackenzie also, she's worked on like marker-based techniques for mice, which you can imagine these markers are teeny tiny because the digits are really small. Yeah, so that was the original motivation. And I mean, now it's just, I mean, it's wonderful. There's so much one can do. And yeah.
This is awesome.
Right on. Yeah. So Amag had an interesting question, which was, are people using this to try to predict behavior based on the patterns that emerge from being able to get good kind of skeletal tracking here?
Yeah, no, exactly. So there are a number of groups that do that. There are also some packages, open source packages that I was mentioning, that are kind of doing that. And we also develop a new package called DLC to Action, which basically is then on top of DeepLabCut, where you can do action segmentation based on key point input or just video input, actually. Yeah.
That's really interesting. I could see that being used for so many different things, even just in terms of animal health care, and just so many possibilities with this. Abkul would like to know how do you think it would perform on a swarm of locusts?
How many?
I don't know. How many's in a swarm? And do
Yes,
locusts have hands?
They don't.
No? Okay. Pretty sure. You got me second guessing everything now, Alex.
Yeah. No, I mean, there is actually, it can be done. I mean, obviously, the question is, let's say if you analyze a swarm of locusts, is your visual resolution really high enough that for each of the locusts you have enough pixels that you can measure, let's say, their posture very accurately? I think that's kind of a key question. In the paper that I can also, our multi-animal paper that was out in Nature Methods recently, we have, for example, swarms of 14 fish, and they're called schools, I guess. And that works extremely well. I think you could easily go up to, I don't know, 30, 40. And there's no principled reason why that's too much. It's just typically, let's say, it's unlikely that the animal has enough pixels that you still want to do pose estimation. You might just want to do multi-object tracking.
Right.
Yeah.
I see. Thanks so much for joining us this week again, Alexander. This was a fascinating look at some cutting edge technology and science, and where science and technology meet is kind of our deal. So super awesome to have you here. You know, we hope that DeepLabCut continues to grow, and it's really great to see the community aspects that you're building up on here, and with the collaborative tools. It's a great addition to the world of AI, and hopefully a great addition to the world of neuroscience and biology as well. Those of you who tune in every week or try to tune in every week, our guest next week will be Jacob Irvin of Active Replica. We may be debuting our fancy new virtual set. You'll have to tune in to find out. Satya Malik should be back with us. Anna, thanks again for being our ersatz Satya. You have stepped up, and I really appreciate you co-hosting the show with me here.
My pleasure.
Alexander, once again, thanks so much for joining us. Everybody out in the audience, thank you so much for watching this. We only do this show because you all watch it. If you have anything you'd like to see on the show, any potential guests, or any other questions from the show that didn't get answered, go ahead and send them to us at newsletter@opencv.org, and we'll do our best to answer them there. Take care of yourselves out there, and take care of somebody else. Thanks so much for watching this episode of the webinar. Please hit that like button, subscribe, and don't forget to tap the little bell icon to be notified when new episodes drop. This week's episode was brought to you by OpenCV Courses. Learn computer vision and AI from the best at opencv.org/courses. If you'd like to be in the audience next week, subscribe to the OpenCV newsletter.